Great, and I'd like to thank the organizers for organizing this symposium. It's been really great to hear about kind of the diversity of approaches in the area to cell biology. And so today I'm going to talk about the work that I've done. So I'm a new faculty at the Fred Hutch. I just started earlier this year. And what I study is nuclear envelope dynamics, and in particular, this dynamic called interphase nuclear envelope rupture. And this is part of this kind of emerging idea in the nucleus field that the nuclear envelope is not the static structure, but actually has a lot of dynamics that are important for its normal functions and could be contributing to disease phenotypes. And so this is kind of the classic view of the nuclear envelope, where it's thought to be this barrier between the cytosol and the chromatin that allows compartmentalization of different functions in the nucleus and the cytosol, such as transcription in the nucleus and translation in the cytosol. And the nuclear envelope has a number of components that make up this barrier, including kind of this double double membrane called the nuclear membranes that are contiguous with the ER. Passage into and out of the nucleus for large proteins and complexes is regulated by the nuclear pore. And then underlying this membrane and giving it kind of shape and stability is the structure called the nuclear lamina, which is what I'm going to be focusing on today. And so the first idea that maybe the nuclear envelope was doing more than just acting as a barrier between two compartments came from studies of the nuclear lamina, which is actually a very complex structure that lines the inner nuclear membrane. So the main part of this structure is the lamin proteins, which are intermediate filaments that form a network on the inner nuclear membrane. But these lamins, in conjunction with proteins that reside in the inner nuclear membrane, have a lot of different functions in the nucleus besides just supporting the membrane, such as they are important for localizing heterochromatin to the nuclear periphery. They're also important for regulating gene expression through interaction with transcription factors. And also there's an idea that the lamins may be important in mechanotransduction, so signaling from the outside of the nucleus to the inside. And this idea is really funneled how we think about diseases associated with the lamin proteins. So there's a large group of diseases called laminopathies, many of which are due to proteins or mutations in this one protein, lamin A. And these are all the mutations that have been identified that cause diseases along the length of the lamin A protein. And you can see that there are a diversity of systems that are affected. You can get muscle disorders such as cardiomyopathies and skeletal dystrophies. You can get peripheral nerve diseases and other forms of skeletal problems. And in addition, one of the best-known laminopathies is this condition called progeria, which causes premature aging in patients who have this mutation. And so one of the big ideas recently has been that defects in or mutations in these lamin proteins can cause a variety of diseases because they have a variety of functions in the nucleus. In addition, changes in nuclear envelope morphology are also associated with the development of cancer. And so these kind of stereotypical changes in either the shape or the size of the nucleus and the nucleolus have been used for a long time by pathologists to diagnose malignant diseases. And so from all of this work, people assumed that the nuclear envelope must be kind of this very static thing, important to be a barrier, and also important for kind of its functions as this platform of signaling. But it turns out that the nuclear envelope can actually be very dynamic in interphase. So there's several different types of dynamics that have been identified, including fusion of pronuclei during early development or of carrier mirrors during early mitotic divisions. In addition, it's also been shown recently that there's a new transport machinery that bypasses the nuclear pore and can transport particles directly into the cytoplasm from the nucleoplasm via budding through the inner and outer nuclear membranes. And finally, we've also seen evidence of nuclear envelope rupture and repair in interphase cells. And this is the process that I'm going to focus on today. So my lab has two big questions. First, what is the mechanism of nuclear envelope rupture and repair? And second, what are the consequences of nuclear envelope collapse in micronuclei? And so today I'm going to be talking about two different nuclear compartments, the primary nucleus, which is what most people think about, and then what happens when chromosomes are missegregated into these micronuclei. Okay. And so kind of as all good cell biology projects do, this started with a stray observation by a graduate student in my previous lab, the Hetzer lab. And he was looking at nuclear envelope reassembly at the end of mitosis using a marker for the integrity of the nuclear envelope, which is just 3 GFPs tagged with a nuclear localization signal. And what you see is that when the nuclear envelope breaks down, the GFP NLS is no longer localized to the nucleus, but kind of diffuses throughout the cell. And then it's only when the nuclear membrane is kind of reformed around the chromatin and that barrier is reestablished that you can now import and accumulate GFP-NLS into the nucleus. And so this is what this looks like in a live cell movie. So this is a PTK1 cell expressing GFP-NLS, and the chromatin is marked by H2BM cherry. So this cell starts off in interphase. You can see that it goes into mitosis, and the nuclear envelope breaks down, and you get the diffusion of this GFP throughout the cytoplasm until the nuclear envelope reforms. And just to give you an idea of the time scale, each of these images is 3 minutes apart. And the whole thing is sped up so that 1 second is about 45 minutes. And so while he was watching this process, he saw something he didn't expect. So this is a U2OS cell in interphase. And using those same markers, what he saw was that these interphase cells were also undergoing nuclear envelope rupture, which was previously thought to only happen during mitosis. And there are some things that you can see about this. First, that the rupture is transient, so the nuclear envelope is able to repair itself and re-import the GFP-NLS. In addition, it's also accompanied by this blebbing out of the chromatin, which I'll talk about a little bit more. And so when I came to the lab, I was really interested in this observation because it seemed like a whole new way to think about the nuclear envelope. So I immediately started developing cells where we could look at this really rigorously. So this is a U2OS cell line. It's arrested in interphase. And what's amazing is that you can see all of these nuclei are undergoing, or most of the nuclei are undergoing multiple rounds of nuclear envelope rupture and repair, and that this complete loss of compartmentalization of the nucleus is non-lethal. So these cells will stay in this state. They can undergo like up to 30 ruptures we've seen, and yet if you release them into mitosis, they'll go on and divide and be pretty normal. And so we're able to find this in a number of different cancer cell lines. So this is just showing a few, the prostate cancer, the HeLa's breast cancer cell line in U2OS. And we see a variation between different cancer cell lines. In addition, we could also see this in immortalized MEFs, but not in some of the other epithelial or endothelial cell lines. And this is consistent with our idea that in cancer cells, they tend to have changes in their nuclear envelope structure that may allow kind of an increase in instability. Okay. And the other thing to point out is that these ruptures, again, are very transient and typically last only about 10 minutes. And so what I've been doing is looking at the mechanism of this nuclear envelope rupture. And I'm going to show you some data now that kind of highlights some of the points that we've seen, which is that in tissue culture cells, the nuclear envelope rupture seems to happen at these areas where the lamina is depleted. And this causes an area of weak membrane that when the membrane is put under tension in tissue culture cells, this is by assembly of perinuclear actin filaments that compress the nucleus, that tension eventually leads to chromatin herniation and then rupture of the membrane. Okay. So first, the reason that we started thinking about the nuclear lamina was, one, because we know that it's important for the structure of the membrane. But in addition, previous work had shown that these chromatin herniations, where we see the most nuclear envelope rupture, are associated with these large lesions in the nuclear lamina. And so what we did was to ask whether or not these lesions correlated with nuclear envelope rupture. And so you can induce these lesions in normal cells by doing a small depletion of the lamin B1 protein. So you can see this is the normal lamina, and then this is the one with all of the lesions in it due to the depletion. And corresponding to an increase in these lesions, we also see an increase in instability and nuclear envelope rupture. And the corollary to this is that if we overexpress one of the lamins, lamin B2, which causes these lesions to disappear, we can rescue nuclear envelope rupture and repair the stability of the membrane. And so this really showed us that these holes in the nuclear lamina were both the site of nuclear envelope rupture and also important for this process to initiate. The second thing we looked at was the effect of the cytoskeleton. So the cytoskeleton is known to interact with the nucleus and to also transmit force through it through either microtubules or actin filaments. So what we did was to depolymerize either actin using cytoclasin D or microtubules using nicotazole and ask what happened to nuclear envelope rupture. And what we saw was that depletion of microtubules had almost no effect on the nuclear rupture frequency. However, loss of actin completely abolished nuclear envelope rupture in our tissue culture cells. And then we asked whether or not this was just the polymerization of actin or something about its organization. So if you treat cells with the myosin inhibitor blebbistatin, it causes disorganization of the actin filaments. And we saw that this disorganization was also sufficient for the loss of nuclear envelope rupture. And so together, these really implied that something about the organization of the actin cytoskeleton was causing the rupture to happen. And we wanted to show, to do several tests that implied that the actin cytoskeleton was actually putting a force on the nucleus that was driving nuclear envelope rupture. And so to test that, what we did was to use a microfabricated device to simulate that mechanical force in the absence of actin. So this is a device that was developed first by the Peel lab in France. And what it is just a cover slip upon which we've printed little PDMS pillars of about 4.4 micrometers. And then we invert this on top of the cells, and that compresses them. And so to give you an idea of what this looks like, in the absence of confinement and in the presence of the actin cytoskeleton, these U2OS cells typically have several chromatin herniations. In addition, if we watch them, they'll undergo multiple rounds of nuclear envelope rupture and repair, shown by the GFP flashes into the cytoplasm here. However, in the absence of actin and in the presence of cytoclase and D, you can see that we don't really see these sort of herniations. The nuclei are kind of blebby, but there aren't these clear changes in DNA structure. In addition, there's no nuclear envelope rupture that happens. And so we wanted to know what would happen if we compressed these cells here. And this is what we saw. So first of all, you can see that there's a rescue of the chromatin herniation here. And then if we continue to watch these cells over time, you'll also see that we are able to rescue the nuclear envelope rupture. Okay? And so this data suggesting that the actin cytoskeleton is putting pressure on the nucleus and that that's what's driving rupture was consistent with other observations of nuclear envelope rupture in more in vivo-like settings. So first of all, there was work from Titia de Long's lab that showed that when you form a chromatin bridge between cells due to telomere deprotection, that when the cells start to pull apart, you'll often see rupture of the nuclear envelope. And in our model, this makes sense as you're putting strain on the nucleus as you're trying to pull the connected nuclei apart. They also suggested that this nuclear envelope rupture was important for kind of allowing in DNases that chewed up the bridge and allowed it to resolve. In addition, recent papers from the Peel lab and the Lammerding lab also show that nuclear envelope rupture can happen in migrating cells in vivo. So these are MDA-MB231 cells injected into a mouse ear, expressing the GFP NLS. What you can see is as the cells are migrating away from the tumor, you can actually watch the nuclear envelopes disrupt and then repair as they migrate away. And so currently, this is our mechanism for nuclear envelope rupture and repair. And at this point, it's very rudimentary. But what we think is required is that you need something that's disrupting the nuclear lamina to generate these areas of weak membrane. But then the tension is actually what's causing the herniation and the rupture. And we know very little about, first of all, why this lamina becomes disrupted. We don't really understand why, even when we knock down one of the lamin proteins, we get these large holes in the nuclear lamina. In addition, we don't know whether or not the actin cytoskeleton is always required for this membrane tension, whether or not cells respond differently if they have pressure coming from inside the nucleus, say due to chromosome decompaction. In addition, we know very little about this repair process. The membrane seems to repair prior to the rest of the nuclear lamina, but we don't know many of the molecular players involved in this process. What we do know is that in cells that have a lot of nuclear envelope rupture, you can see mislocalization of both cytoplasmic organelles to the nucleus, such as mitochondria. And you can also see mislocalization of nucleoplasmic organelles, such as PML bodies, to the cytoplasm. So we think that this can have really significant consequences for the cell and for the nucleus, but we don't understand what they are yet. And so in the second half, I'm going to talk about the work that we've done looking at nuclear envelope stability in micronuclei. And so in mammalian cells, micronuclei arise from defects in chromosome segregation. So typically when you have either a chromosome that's lagging during anaphase or a chromosome that can't form a kinetochore and can't align on the metaphase plate, what happens is that if it's far enough away from the rest of the chromatin mass, it'll recruit its own nuclear envelope, leading to a G1 cell with two nuclear compartments, the primary nucleus, which contains most of the genome, and the micronucleus, which contains either a single missegregated or a couple missegregated chromosomes or a chromosome fragment. And so micronuclei are very common in cancer cells because they typically have defects in segregating their chromosomes. But if you take a cheek scraping right now, about 1 in 1,000 cells are going to have a micronucleus. So this is also something that happens in normal tissue. And so while we were looking at our cancer cells, we noticed that something interesting was happening to the micronuclei. So this is, again, a U2OS cell expressing GFP-NLS and H2BM cherry. You see this cell is just divided and formed a micronucleus and a primary nucleus. And if you watch the micronucleus, what we saw was that at some point during interphase, the nuclear envelope of the micronucleus would rupture. And unlike the primary nucleus, it wasn't able to repair. So this chromatin would kind of stay together and sit in the cytosol until the next mitosis. And this was a very frequent fate for the micronuclei. About 60% of them disrupted every cell division. In addition, this was very consistent between different cell types. So most of the cancer cell lines we looked at, about 50 to 60% would rupture. And if we induced micronucleation in normal cells, we also saw the same level of nuclear envelope rupture without repair. And so we're really curious about why the nuclear envelope wasn't repairing. And I'll tell you right now that we still have really no idea what's going on. But we did see some interesting changes in the structure of the membranes and the chromatin after nuclear envelope rupture in the micronuclei. So this is a cell expressing now RFP-NLS. The DNA is marked in blue, and expressing an ER protein tagged with GFP. And so this cell has a primary nucleus and a large micronucleus. What I hope you'll notice is that when the micronucleus disrupts, right there, you can see already that at this point, the membranes are excluded from the chromatin. But as we go on, the membranes kind of start invading the chromatin, and the DNA looks like it's collapsing. And we can also see this if we look by EM as well. So this is the primary nucleus and the micronucleus. You see the chromatin looks pretty similar. They both have this membrane surrounding the chromatin. However, in the disrupted micronuclei, it looks like the chromatin has become massively compacted. And also you can see ER tubules have now started to invade the chromatin. And at this point, we don't know whether or not this is a cause or consequence of the lack of nuclear envelope repair, but it's something that we're planning on investigating. Okay, so what are the consequences of losing compartmentalization of the nucleus? Well, they're pretty much what you would expect, in that most of your nucleus functions are going to be lost. So we see that in the disrupted micronuclei, that they're no longer able to transcribe mRNA, whereas the intact ones are. And this is quantified here. In addition, if we look at DNA replication by EDU incorporation, we can also see that the disrupted micronuclei are unable to incorporate EDU, indicating that they are unable to replicate their DNA, which is shown here. In addition, to our surprise, we also saw that the disrupted micronuclei seem to have a lot of DNA damage, as indicated by the gamma H2AX staining, which is a marker of double-stranded DNA breaks. So this is a cell with an intact and a disrupted micronucleus. And you can see this accumulation of gamma H2AX on the disrupted chromatin. And again, this is very specific to nuclear envelope disruption. And we were able to show, using some timing experiments, that this damage is really associated with the act of disrupting the nucleus and possibly with compacting the DNA. And what's been really exciting is that another lab, the Pellman lab, has gone on to show that this disruption of the nuclear envelope in micronuclei and this DNA damage can actually be the source of massive genome rearrangements called chromothripsis. So they were able to follow 9 cells by live cell imaging. And in 8 out of the 9 cells, they saw that the chromosome that had been micronucleated now had all of these rearrangements on it. So the idea is that the chromosome got shattered, and we can actually see these fragments in mitosis. And then the next cell cycle, it got stitched back together. And so we know that this chromatin can actually stay together during mitosis. So we can take cells that have a disrupted micronucleus, follow them through mitosis, and what happens is that this DNA is either going to go into the primary nucleus and segregate with the rest of the chromosomes, or it's going to end up in a micronucleus again. And if we look for how many cells look like this and don't have a micronucleus, we can see that in both the intact and disrupted micronuclei, that chromatin is rejoining the rest of the genome about 50% of the time. So this DNA isn't being lost from the cell, but it's persisting at least into the next cell cycle. And then at this point, we had done everything in tissue culture cells. So we wanted to know whether or not this was also happening in vivo. So what we did was to look in tumor biopsies from patients, and this one is a non-small cell lung cancer, particularly a squamous cell carcinoma. And using our markers of the disrupted micronuclei, including gamma H2AX, this is just using E-cadherin to allow us to outline the cells, we're able to identify cells in this tissue that have a primary and a micronucleus, and where the micronucleus is disrupted. So we know that this is happening in vivo. In addition, recent work from other labs has shown that when micronuclei form during early embryogenesis, such as in mouse embryogenesis, that you can also get disruption of this nuclear membrane. And so we think that these two occurrences may be contributing either to the development of cancer or to the development of birth defects from aneuploidy. And so when we think about kind of the overall consequences of nuclear envelope disruption in micronuclei, there are a number of ways that it can be affecting the genome stability of the cell. So first of all, when you lose transcription from this entire chromosome, basically in interphase, the cell is going to be 2n-1. So it's going to be experiencing this temporary aneuploidy. However, if this DNA was not able to fully replicate before it went into mitosis, then you're going to have a problem with your chromosome number, where again, one of the daughter cells is going to be 2n-1, so kind of a persistent change in the ploidy. And finally, we know that if this DNA gets reincorporated into the primary nucleus, if it's been shattered during mitosis, it can also be reassembled into these highly rearranged chromothripsis chromosomes. But in addition to all this, we think that even in the absence of chromothripsis, that just being in the cytosol for that long changes the structure of the DNA and changes the histone modifications. And so one of the major questions in my lab is, what are the other consequences of having this disrupted nuclear envelope for the chromatin? Is it able to regain its ability to transcribe efficiently? Does it have other types of changes in gene expression that we can follow? And so right now we're trying to develop tools to follow this DNA through multiple cell cycles and ask what the consequences of decompartmentalization are. And so with that, I'd like to kind of acknowledge where I've been. So a lot of this work was done at the Hetzer Lab at the Salk Institute with help from the UCSD Biodynamics Lab, and also a collaborator at Cornell who provided a lot of reagents. And the Hatch Lab is now 3 strong at the Hutch, and we invite anybody who wants to come by and talk about nuclear envelope structure to do so. So thank you. Any questions?
So in the morning we heard about high-seq and back-seq.
Yeah.
Do you foresee any chromatin differences before rupture and after rupture?
So we haven't looked particularly at that. So we know that in the primary nuclei, that the chromatin that tends to be in those herniations is mostly euchromatic, because we think it's the one that can flow out and form those blebs. And we haven't looked at how that affects it. With the micronuclei, it's actually a really interesting question. Because when you think about it, that chromosome in the micronucleus may not have contacted the nuclear lamina in the main nucleus. And now it's having all of these different sorts of associations with the nuclear lamina. And so we are really interested in trying to purify them and see if there are changes in the chromosome conformation in addition to DNA replication and kind of other things like that.
Hey, Emily, a great talk. So at ASCB, I saw some posters where people were forcing migrating cells through small channels, and then it was rupturing the nucleus, and then they showed that escort was repairing it. And so in your micronucleus, maybe you haven't looked yet, but I mean, I wonder, why doesn't the micronucleus repair? And then if you could look at like whatever's upstream of escort, is that not getting recruited?
Yeah.
Like
Right. No, we definitely want to look at the localization of escort and whether or not it's getting recruited to the sites of the micronucleus rupture. At this point, we don't know. In addition, I don't think anybody really knows what's upstream of the escort when it goes to those migrating cells. So we're hoping kind of to develop screening techniques to identify those players in our system.
Yeah, on that note, I was, so one issue when you have the micronucleus and then a giant nucleus next to it that's intact, maybe in some way that's outcompeting this process in the micronucleus. So I'm wondering, have you ever tried to rip a hole in the large nucleus at the same time that you have the micronucleus and seen whether it affects the ability of that micronucleus to repair?
Yeah, so we haven't looked, well, we have one movie that suggests that maybe not. So we have a movie where the micronucleus disrupts and the primary nucleus starts rupturing a whole bunch, where, all of its basically soluble contents are now in the cytoplasm and presumably available to the micronucleus. And in that case, we can see the proteins kind of get a little bit recruited, but then they dissipate. So it's not able to repair its membrane in that situation. But again, at that point, the chromatin structure may be different. The membrane structure may be different. Like it already may be repair incompetent. But we don't know if, right at the time of rupture, if it just needs additional factors. Great. Thanks. Thank you.